Howdy folks, thanks for checking out the AP Biology videos. I am Mr. Ulrich. In this video we're going to be looking at the cytoskeleton and how cells connect to one another. The cell's cytoskeleton and your endoskeleton have some of the same basic functions. One of those functions is to fight gravity and kind of hold the cell in a particular shape. It's also going to hold the organelles in their right spot, most notably the nucleus. And you can see in this picture that just came up how there's quite a bit of yellow framework. We'll talk about those types of structures as we get on in the video. The other thing that cytoskeletons do, same thing as your endoskeleton, is it does provide a rigid structure for leverage so that the organism can move itself and to move material within the cell. With that in mind, the cytoskeleton is also involved in regulation since it does move things like the chromosomes during mitosis and meiosis, as well as cytoplasmic streaming and cyclosis, moving the material around within the cell. The structure of the cytoskeleton involves fibrous proteins extending throughout the cytoplasm. There are three different protein fibers that we should be familiar with. Those are the microtubules, which are the big tubes, the microfilaments, which are the smaller kind of thready bits, and the intermediate filaments, which are the branched guys that really are holding lots of stuff together. The basic way to tell these things apart is by how big they are. And this diagram gives you a little bit of a picture as to the relative size of the different things. Here you can see that the microtubules are pretty large in comparison with the microfilaments. The intermediate filaments are kind of midway in between the two. So big, medium, small. When we start comparing the cytoskeletons of different types of cells, we see that there are quite a few homologies between the different proteins that make up the cytoskeleton, even from things as distantly related from bacteria and humans. Prokaryotes and eukaryotes do show quite a few similarities in these proteins, like the tubulin that's common amongst all organisms, and actin, which we see in eukaryotic cells. These are old, they have strong adaptive values to the point where they are essential. Let's start to take a look at each one of these fibers a little bit more detail. Microtubules are the big ones. These are the honkers. They are hollow, so that's a nice, it gives them good structural integrity. So they're hollow rods made of a protein called tubulin, and as the tubulin dimers are added, you can make these fibers longer and can make them shorter by breaking them down, which is readily done in the cytoplasm. The function of microtubules extends beyond their structural kind of strut function, holding the cell up or giving it its shape. They're also involved in moving materials throughout the cell. The chromosomal migration during mitosis is dictated by the construction and destruction, really, of microtubules. The centrioles are really the organizing structures of those microtubules. They also act as kind of like railways for motor proteins to actually walk along. When you see this animated, it's a pretty cool process. These dyneins and kinesins, they're motor proteins. Myosin is another motor protein of sorts that interacts with actin, but it'll also interact with tubulin as well. Anyway, the way these motor proteins work is as they get phosphorylated to cause a conformational change in the protein, which causes this thing to actually take a step forward. Kind of cool. These microtubules do have a directionality, a positive and negative end. So these motor molecules, motor proteins, do have a direction that they're heading in. The microtubules also function for motility. They create the cilia and flagella that are going to power the cell, move the cells around. Um, Not power the cells, that's going to be cellular respiration, which we'll talk about later. But the cilia and the flagella beat the extracellular environment in order to either move the environment, in the case of cilia that line your trachea, or to move the whole organism or the whole cell, like paramecia or sperm cells. Centrioles, like I said, are the organized structures of microtubules. They are involved in cell division because they are involved in creating those microtubules, although their involvement is not very well understood. There are cells that divide just fine that don't have centrioles. We only see these in animal cells. In each animal cell, there's a pair of centrioles. One is called the mother centriole, and the other is the daughter centriole. And they divide on their own in order to, like I said, guide the movement of chromosomes during mitosis. They also form the basal bodies, the anchor structures, basically, for the cilia and the flagella themselves, made out of microtubules. Speaking of cilia and flagella, these really are just parts of the cytoskeleton that have gone through the cell membrane and extend out into the external environment. If there's lots of them, and they're really short, like on this paramecium, we call them cilia. Flagella are longer. You typically only have one or two per cell, though some cells do have more. They do work, both of them, by either moving that organism through the water by swishing the water by them. Sometimes, in the case of the cells that line your trachea, they actually move the environment while they stay stable in one position by sweeping that material in one. Cilia move like little oars. They kind of row back and forth. They generate the power perpendicular to the direction that the cilia is beating. So when the cilia are beating back and forth this way, the organism is moving this way. So the cilia stick out this way, the organism moves this way. Flagella are the long ones. They move in almost like a circular motion. Um, and in this one, the flagella kind of points in this direction, moves in this plane, and the direction of travel is in the same plane. The structure of cilia and flagella, we used to have to remember that they are made up of 9 little double microtubules surrounded by 2 in the middle. It's less of an issue that you have to have those 9 pairs of microtubules around 2 single ones in the middle embossed in your brain. But it is cool to know that they are made of bundles of microtubules. The bending of them is driven by that motor protein that we were talking about before, dynin. In this case, they're attached to one side and they kind of walk along the other side. And so when ATP is used, that conformational change actually causes these 2 microtubules to slide past one another. So one moves in one direction, the other moves in the other direction, and that'll actually bend the whole structure. When we look, the basal bodies have a slightly different arrangement. They have their microtubules in triplets, and there's no central ones, as opposed to the cilia or flagella itself that have, here's the doubles, 1, 2, 1, 2, right? The 2 single ones in the middle. So there's 9 plus 2, and this is 9 triplets. Well, the microtubules are the big guys. The microfilaments are the little guys. They're made of actin. You'll probably remember hearing about actin and myosin. That actin forms a double helix. The job is to, like the microtubules, to give kind of a 3D shape to the cell, but they'll also interact with motor proteins to contract, so not just in muscles, but also to contract the whole cell. They work by constantly forming and dissolving. So you can kind of give the cytoplasm a little bit more stiffness or make it a little bit more liquid. And so by changing where it's stiff, make it stiffer on one side and more liquidy on the other, and then also doing some contraction, you can start to move the cytoplasm. So the pseudopodial movement of amoebas and your white blood cells, for that matter, comes from the action of these microfilaments. It also maintains a fluid nature to the the cytoplasm, so it does influence the cyclosis in plants. Intermediate filaments, these are the ones kind of mid-grade, mid-size, not mid-grade, they're actually really, really strong. These guys are built of keratin, just like in hair and nails and beaks and stuff. The function of these guys, they're a little less dynamic. Their job is to really hold things, the more permanent structures like the nucleus, in place and give that cell more shape, just like the rest of the cytoskeleton. So just to kind of wrap up these three different types of tubes, the microtubules are the big ones. They're used for, with those motor proteins, the dyneins, for moving materials around within the cell. They're also involved in moving material, making the cilia and flagella, so they're also involved in motility of the cell. And they're made of tubulin. The intermediate fibers are intermediate in size, that's helpful, and they are for holding more permanent structures in place. They're made of keratin. The microfilaments are the tiniest ones. They're involved in cytoplasmic streaming, as well as phagocytosis and endocytosis and all those other good cytoses. Um, and they're made of actin and myosin. All right, so now we're getting closer to identifying all of the structures within the cell. Now we've covered all those organelles, the endomembrane system, the centrioles, the cytoskeleton, a lot of good stuff. We need to recognize, of course, that, like it says down here, we should be able to identify what structures are not in the animal cells. We'll get to plasmodesmata in a little bit. And here's the plant cell, and again, we need to be able to compare this to the animal cell and know that we don't have lysosomes or centrioles or flagella, though sexual reproducing plants do make flagellated sperm in some cases. All right, now we got to talk a little bit about the places where cells get together, where they actually meet. Here we're talking about we're gonna focus on plants and animals, though funguses do have some interesting cell junctions. That is for sure. Plants, being good multicellular organisms, have ways of gluing their cells together using their cell walls. Structurally, it is made of cellulose and has about three different layers that we need to talk about. There's the primary cell wall, which is basically makes up the outside of an individual cell. There's a secondary cell wall that's kind of inside of that. So on the inside of each cell is a secondary cell wall. Between the two cell walls of individual plant cells are the middle lamella. Within the middle lamella are sticky polysaccharides that really glue those two cells together. Being a cell in a multicellular organism means you have to be able to share material between different cells. So there are intracellular junctions, places where neighboring cells kind of meet. In plant cells, these are called plasmodesmata, and these are little holes that actually make channels for cytoplasmic material to be able to pass between neighboring cells. The external environment of an animal cell is quite a bit different. We don't have the cell wall like plants and funguses and a lot of protists do. Instead, we have this extracellular matrix, which is largely collagen protein fibers shot through a forest of glycoproteins. Glycoprotein is a polysaccharide molecule that's attached to a protein molecule. Their job is to provide kind of the same, in some of the same ways, the support that a cell wall would provide, giving the external strength to the membrane itself, to the outside of the cell. They also allow the cells to stick to one another, and by doing that, they create points of leverage, so that allows multicellular organisms to coordinate and move. They're also involved in regulation. They allow these polysaccharide molecules are what your immune system uses to recognize self versus non-self. The intercellular junctions in animals are a little bit more complex than they are in plant cells. There are three different ones that we should talk a little bit about: the tight junctions, the desmosomes, and the gap junctions. The tight junctions themselves, these are, just like they say, these form kind of impenetrable barriers. These are going to allow cells that are next to one another to make like waterproof barriers or airtight barriers, so nothing can get past them. In between the cells. You can't get stuff in between the cells. That means that the only way that you can get material into the or past this line of cells is to go through diffusion or active transport and actually go through the cells themselves. Now, gap junctions, these are spots that actually allow the cells, just like the desmosomes in plasmodesmata, excuse me, just like the plasmodesmata in plant cells, they do allow for movement of material between them, between neighboring cells. But it's tightly controlled by these connexin proteins, which are pretty cool, and they change shape and rotate and open up like the iris diaphragm or a shutter on a camera. And these do allow the exchange of material, so you can get ions and signaling proteins and all kinds of stuff to go through these gap junctions. Desmosomes, on the other hand, these are places, again, points of attachment for cells to stick to one another. They're made out of keratin, and they're what allow these groups of cells to fit together into big, wide, strong sheets. Well, we'll stop there for now. Thanks again for checking in. Again, I'm Mr. Ulrich. You can get lots of supporting information at the website, mrulrichslandofbiology.com. If you have any questions, comments, concerns, or anything else, feel free to drop me an email, and I'll see you in class.